And I want to make sure we have screen share capabilities. Yes. So I think with that, Professor Lau, I think you should be able to take it away from here.
Hi, can everyone hear me?
Yes.
So hi Ethan, hi Bina, hi everyone else. Yeah, so just as a heads up, I try to do it alone, but there might be any minute where my son will wake up and then we have to co-moderate this. I will just, you will probably hear the when the time starts, but I'll try to move ahead a little bit. So let's do this. So you can see my slide with the title NDD-CNV Portal. Okay, yeah. So yeah, it's an interesting time now. So at the moment, people don't want to hear about my science. So and just to show always these shops of data, which is good. So so I'm having a little bit unusual perspective, probably similar to Ethan, which is a little bit bird's eye perspective on the field. So as a genomicist, I'm so-called like a gene or CNV hopper. So we work on many projects and try to understand disease as a whole. But sometimes really it's really useful to see where often redundant tasks are done. So where if you complete the task for one gene or for one genomic region or for one CNV, you could actually do it with 5% more effort for additional disorders as well. So you don't need to hire a new bioinformatician or develop a new or reinvent the wheel twice. And this is what we try to streamline. If we figure something out, we try to develop online resources that hopefully can then help moving other projects forward and focus really on the task which cannot be repurposed, basically. So, and I will soon talk a little bit more detail, but just for everyone who may be really involved in detail, for example, with chromosome 8p or chromosome 15 or ring 14 or other copy number disorders, I think one thing which is a different problem for single gene disorders is that we actually have a fairly good understanding what each gene does and if it's getting affected by, and if it's completely removed or added to the genome. So individually, this is in particularly for disorders like 8p, not the main challenge. So it's not, the gene level is not the problem. Typically, the gene level is the problem. It's more the problem what the combination of genes do together. And if you remove them together and in which combination you do this, what is the resulting consequence? And to me, I think I tried just to think before this meeting a little bit how I would describe this. But I guess it's a little bit like if someone would try to shoot you, or like 10 different people would shoot you, always aiming for the heart. And probably every time the person would shoot a little bit different, so the bullet would have a different entry. And depending what the bullet hits, you would get different organs affected, and the outcome would be slightly different. And the problem is we don't really know, what entry and the combination of organs in this, would definitely yield to the disease. We know what, if we have a good understanding what each organ does if we would hit that in isolation. Not some better, some others not so much, but the combination of it makes a problem. And this is harder to predict, and this is the challenge what I think we face here, and particular for disorders like 8p, where you have different combinations of genes or organs which could be hit. Okay, so now let's move a little bit more detail into this. So we developed this online resource throughout the Neurodevelopmental Disorder Commission project, which we did over a year, where we met every month, and we're thinking how we can try to bring in some data which is currently disconnected. And this is also, a thing what I feel that which is happening more and more. Or we come more and more to the same conclusion. So what I'm talking about. So even for many monogenic, we know which are considered as easy disorders, we have the situation that, and this is not different for the copy number, that if you ask a clinician how many versions of the disease are there, one clinician might say 4, the other one say 3, and so on. And at the end, if you would ask 1,000, this was average out on, let's say 3.5. So, but there's different opinions, right? So, and that there is no consensus point, right? So there's no agreement from the scientific community that says it's 4, right? No, this is still very much research. And different levels of expertise have different ways of looking at this. And you can also define disease in different ways. So there's no consensus. There's no consensus statement. This is also true for 8P, for example. So and if you would ask biologists, how many versions of the disease are there? How many mechanisms are there? Even with a single gene, you might get an answer from people who do one type of experiments. Let's call it electrophysiology. They would say there are 3 different versions. We can measure that 3. But people who work with mice or do some other kinds of behavior assessments, when they see, even in humans or in animal models, they would say, yeah, there's basically 2 groups. And again, there's no official statement consensus agreement. It's a matter of opinion. So, and if you go through all kinds of ways how we can look at a disease or a disorder, we don't know the truth. So, and this is the problem. So, and in the absence of truth, we can only make educated guesses. So, and this is now why we do these portals. We try to make, we provide all the resources that one can make the best educated guess on on multiple factors, from the clinical spectrum, from the molecular spectrum, from different types of experiments, and so on. So, and this really requires that we have more data. Of course, with more data, we can hope that there is more agreement. There might be, for example, if the clinicians say, and clinicians is a broad term, could be epileptologists versus, I don't know, behavior therapists, there could be also different levels. So, but if I believe there could be situations where one would say, okay, there are 3 groups for certain, and multiple levels of clinicians and even molecular data would support that. And there are 2 different versions which we are not so certain about. So that could be that's unclear, but these 3 versions where we have on clinical level, on electrophysiology level, on who knows, chromatin, whatever level, here we agree on. So, and we need to get to that level. So, and we are not there yet for many, many disorders. And so, but without clear definition of the disease, it's really challenging to go into clinical trials, because probably even every version of the disease needs specific endpoints. Might be the case, might not be the case. But we are really, even for well-studied monogenic disorders, we are, I think, more behind than we realize as a community. So, but still, if biotech develops drugs, do we stop testing them, or do we stop, or do we now give up? No, of course not. And this is now the direction where we are moving, and this is the direction of enrolling based on guesses. And this is very similar what is done in epilepsy surgery. So where you know that you will likely fail 3 times of 10 from 10, like 70% will work, 30% will not work. And you do something which is super invasive. You cut a large piece of brain out of a patient's brain. So, and it's done on a daily basis, right? Because the benefit overall is greater than not doing anything, and for drug-resistant disorders. So, and this involves a lot of ethics, a lot of but overall there's a consensus agreement. There's agreement that people should investigate it if surgery is an option. And even though you don't have 100% guarantee that it will work, and there is a significant fraction where it's not work, people will go for it because there is a benefit there. And this is where we are moving towards in this area in precision medicine, right? So And therefore, again, we need to have more and more information which can be incorporated in decision-making for enrollment in clinical trials. So, and here's now, you know, in a computational way, how to bring a lot of data. So, because imagine we would have now a clinical trial for a specific version of 8P disorders coming next week or so. We cannot expect that every clinical center can make this comprehensive assessment, looking through the literature, doing bioinformatics and so on. That will not be the way. There needs to be resources, how to, you know, what kind of characteristics an individual must have to go for drug 1. And there might be different characteristics for drug 2. And for heterogeneous disorders like 8P, where the boundaries of the CNV are not always the same, it will make sense to have resources to do an assessment. That's the long shot goal. So here's now the portal, and where we have also some information for individuals who are not familiar with you know, structural variants or chromosomal disorders, but I don't need to talk about this today much. And then here's now the interface for the chromosome viewer. So on top you see the chromosome band, so where we are on the chromosome, and then you see a wide set of tracks, and I will talk about these tracks like in a little bit. So, and so if you now move to the beginning, so you see here now the chromosomal region. So this is the P arm of chromosome 8. So, and we have this interface where you can zoom, and I have the link on the last page if you missed it so far. So you have, and also I think the group can just share it around. Yeah, the presentation will also be recorded. So okay, so and then you can look at this region, but you can also just type it in where we are, to then learn more about a specific region, and you can extract this and you can change the region, and you can also see it when you just add the information now here in the next step, either on chromosomal bands like this 8p21, or you just type in what you see in the genetic test where it starts and where it stops. So there's relatively straightforward way to query a specific region of interest. So the next thing is what you see then is within this interval here, which you saw here on the top, you can, so we are now in this part of the chromosome, and this is exactly from, I guess it's 20 million to 34 million on chromosome 8, the region, and you see also all these genes which are here. And these genes are colored by some, for basic scientist relevant matrices, to which assign a little bit of importance about each gene. And you can read about this. We have them, these matrices where they are indicated here in this info field, and we describe then why they're important. And there are matrices which say with a gene is more sensitive for duplications, and some matrices say the gene is more sensitive for deletions. And some genes are both. Actually, most are both. And this is more relevant for like biologists who want to focus on specific genes in that region for specific molecular, to test specific molecular hypotheses. So the main point I want to make in this presentation is here we have a gene track, but for everyone who works on transcriptome, chromatin, or who knows type of genomic annotations, we could show here more, you know, annotations. At the moment, it's just genes, and we know that genes are not explaining everything. So, and if you have a genome-wide track, like a BAT file, it takes us minutes to add this to this table, and we could work with you together to figure out what is the best way to show it also in table format. So, but I come to this a little later. But this would be one suggestion where we could improve, just add more tracks to, in addition to the gene track, we could have a chromatin track, for example. I think that would be probably the next, or methylome, or who knows. So anyways, but I mean, just here to give some ideas, and you are the experts, you know best what would be meaningful. Okay, so we are still within this gene track, and we have now here also this table format, because sometimes if you have a large CNV, you have like 50 genes or something, and it's hard to just capture by eye. And you can now see in this table which genes are, for example, associated with diseases, and what other databases telling us about these genes. So So on the one hand, you saw once I clicked here on this gene, we end up in OMIM. So this is here, for example, here's now the gene FP2. And here, this is like a very famous database, which tells a little bit about the molecular biology of that gene, but also about what we know about this gene clinically. That's one of the NIH oldest disease databases, which is very well curated in terms of functional data. So the next step is that we can look at just all genes, and you can see here already that this interval, that there are like 152 genes in total, and of these, for example, 17 are known to be sensitive to deletions. And again, on this question mark track, you can read about where these, how this love score is generated and which publication is that from. So, and you can see also there's other matrices, and here, for example, there's one gene sensitive for triplications, so duplications. So, and how this score is generated, you can also read about it if you click on this question mark. So that's one way to, if you just want to look at those genes by how they behave through evolution, that's how these scores work. That's one way to prioritize genes. But the more easy way to prioritize genes is just looking at those 152 genes which are known to cause disease. And this is just the next track here. If you click on this ClinGen track, which looks at these 152 genes in this interval and just looks at how many do we know are disease genes, or which ones are these. And this is then the next step. And we see we have 11 disease-associated genes here. So, and you can then look, what level of certainty we have for this disease association, because we take this from the so-called ClinGen consortium, which is from the NIH, the largest clinical, group of individuals who is vetting our genome and how much we know about the gene to disease validity. And you can see now, okay, that the ones which we're looking here, these none of them has really strong evidence. And you can see also the last time this evidence was taken for association with disease. And you see the mode of inheritance, and you can see also which disease has been associated with it. So it's relatively straightforward to look at a large region and then scroll to those which have the classification proven. So then you directly see the obvious in a few seconds. Okay, so, and yes, so for example, we have in this region now several genes, but here these are recessive ones. It's probably not relevant for us because the deletions are typically seen as a heterozygote. So in this case, not here's definite, also recessive. So this is, that's why it's important to carry on this information, not just if the gene is associated with epilepsy, but also in which, inheritance pattern. In this case, if most of these deletions will probably be heterozygous, so dominant, they would not be considered as pathogenic. So this information is here, and you can do the assessment here and work with, for example, a genetic counselor or a biologist. And I think biologists should be able to interpret this and then filter this region very fast. So, and for me personally, just combined this known disease list with also the unbiased list. So the known disease gene list is based on knowledge from humans in the literature, whereas these gene annotations, which we looked before, this is generated through scores, where we can see that certain genes don't have stop codons. So this tells us they cannot handle deletions. So this is a little bit more from a bioinformatics, genomics perspective, and this is computationally generated, these evaluations, whereas these ClinGen interpretations are generated from scientists and clinicians. So in both ways can help each other. For if you have agreement for both, for example, it's associated with a loss of function disorder, and you see also that the score for loss of function is high, then you have a good candidate to follow in your experiments. Okay, so let's see next. Okay, so on the next thing, what we have here is we implemented like a gene set enrichment analysis. So because many of these CNVs affect many genes, and sometimes the question is if they affect a specific pathway. Oh, sorry, I'll start here again. So for example, if we select, and we can do many gene set enrichment analysis, and we did now here one for HPO terms, so human phenotype ontology terms. And we see that for some reason, there are 14 genes known associated with a specific skin nodule. And we see 3 of those 14 genes are in this interval, what we looked at on chromosome 8p. And this is, yeah, it's significantly enriched in that region. But to be honest, this p-value is not very strong. So it's 10 to the power of -2. So typically what I like to see is something more like this here, 10 to the power of -8 or so. That means that, you know, it's very, very significant that this association did not happen by chance. And you can see clearly here, if we look for rare disease, we find that this is has an association with chromosome 8p CNV disorders. So in this, you know, if you wouldn't know this, we would now very easy find now the described disease for that region. So, and you can play with this. There are now many annotations here, where you can just select a region on chromosome 8, or you can actually do this genome-wide. So that tool works for every chromosome, and just see if there's an unusual density of genes which do something together in that region, and that might inform some hypothesis. You could imagine that, for example, if it's at a specific area, then you suddenly something comes up with a skin disease in the left side, and you see also like a skin nodule, and that might explain why some patients have a little abnormal, you know, skin. I don't know. So, but here are ways to develop some hypotheses. So, and this is now for the biologists. So, as I mentioned before, when we have for this gene type of analysis now these kind of ways to query the genes in a different region which we selected, but there could be many more tracks. There could be transcriptomic data, which this is to some degree in this gene set enrichment analysis, but could be done better. And there could be protein-protein interaction type of analysis. There could be, again, methylation, chromatin, mouse knockout screen cell line data, you know, which could be available from patients, some kind of organoid data, who knows. So everyone who generates models and does genome-wide readouts or readouts for the region of 8p, in the chromosome arm 8p, we could incorporate that here and make that explorable. So just as a comment where how this could be improved. And so the next step is the patient track. So here we actually took the data from Wendy Chang, which was published earlier last year. And we look at a specific region. So here's, for example, the region on chromosome 8. So it's the same one I had selected before. And we can filter now by clinical annotations. So because for Wendy's paper, we have all the names, not names, all the patient IDs. And then we have information if patient 1 has seizures or not, or if how old was patient 1, for example. And what we can do now is we can select here now in the bottom different clinical information from that list, which is basically the list of information which was in Wendy's paper. And we can see now if if there would be a pattern. So we can see we lose a lot of, so each bar represents a patient CNV, and we see we lose quite a bit. And you see also that here in the middle actually we lose quite a bit. And this might help to understand if there's a specific region which is only associated with one type of disease. So, so once we can do this just manually like this, or we can just select the region and can see if there are, for example, differences in patients with, for example, deletions or duplications. And here, for example, we don't see much of a difference. So each dot represents a CNV. So, and we can also see if a specific region is associated for a specific type of genes. So if they're deletion sensitive or duplication sensitive. So again, here, so for example, here what we see now is the age, because I selected age here in the bottom. I think my hand is not, yeah, okay. So I selected age, its clinical feature. And you can see that deletion and duplication carriers, maybe the inversion duplications might be a little bit younger, but here you can do so-called like genotype-phenotype correlation. We can explore if a specific region where you might have a hypothesis, maybe there's a gene which you're interested underneath, or maybe there's a specific pattern. You can explore if the age of the patient is changing. And that could be also people having an MRI positive or negative, or that could be degree of intellectual disability. If the more data we have, we can generate these kind of visualization, and you can then basically browse through the genome to test specific hypotheses. And you can see how it changes if we change this region. Here's this region now selected, now it's here. And now we move over to select this region, how then the data changes. Yeah, so we suddenly have more CNVs. Okay, so, but there, so here's no age, but there might, this can be done in many ways. So again, can like have movement disorder problems and so on. It can be done, you know, in many ways. So long story short, so with more patients, we can actually do this browsing through the genome automatically. So we don't need to browse any, you know, you don't need to browse as a human being and see if there's an enrichment for specific phenotypes in specific areas. We can do this also with patients with different needs and specific chromosomal regions. We can do this automatically. And then we can identify is there a region which is more likely to cause severe autism compared to mild autism, or drug-resistant seizures versus mild seizures. There are many ways, but it needs more patient data. And also in addition, we need you know, better phenotype data, so clinical data, which have been organized in a standardized way. At the moment, I think a lot of sources of data come from different centers and from different clinicians who might have different interpretations of the data. So, and not much detail. So the better data we have, we can do better this type of genotype phenotype analysis. So the story I want to, you know, also the point I want to make is like really genotype phenotype algorithms are feasible, and they might really inform, you know, biologists what to focus on when they generate their models, but we need more data. That's it. So, and just as a note, we generated these results based all on public data like a year ago, or like almost 9 months ago. And we don't have much more data today, right? So that's a little bit of the slow progress, but I hope that will get better. And the more people share, then, you know, better we can build better resources. Okay, so the next part is where we have now further down. So it's all the time the same region, right? So what we have selected, we can look now at CNVs which have been observed in the general population. So that means like people like you and me who don't have a neurodevelopmental disorder. So and you can see here that most of these regions here don't have, if you look at 500,000 individuals from UK, just general population, so not with a neurodevelopmental disorder, we see that most of these regions in this 8P interval don't have CNVs in people without neurodevelopmental disorders. Unless there's a couple regions like this one, or this region. Here we have quite a bit. So if you would, for that reason, we as geneticists will conclude that these smaller regions, they are not relevant because, or not relevant for neurodevelopmental disorders, because way too many healthy people without neurodevelopmental disorders have a copy number variant in this region. So that can be used like as a negative filter if you want to for prioritizing genes in this interval. So the opposite basically, but again, this is all the time matched here. So if this region, you can see this here also nicely. This is from 500,000 people from the general population. And you see the same region has actually a drop for now patient variants. So this is now a track where, so from the disease database ClinVar, where genetic counselors had to make the decision if there would be a CNV in this region, if they consider this as pathogenic or not pathogenic. So, and here's now like a density plot where you can see here they would typically say, yeah, it's not pathogenic, or at least, yeah. So, but these CNVs are very large, so it's they probably only make this decision here for the small CNVs, and you can just count now how many patients CNVs we have now, which were classified as pathogenic for deletions and duplications separately in that region. So what we could do more here is add more disorder-specific genomic tracks. For example, there's a paper where I played also a small role, where for 1 million people, including 280,000 people with a neurodevelopmental disorder or psychiatric disorder or neurological disorder, the genome was scanned for regions where you have in patients more CNVs compared to people who don't have that particular disorder. And they looked at, I think, 40 different disorders, including, I think, 21 which are brain related. And we could always display like tracks like we saw, which we saw here. We could display these kind of tracks which for every of the 21 brain disorders across this region and see if there's a peak, for example, for this is an autism region or something like this, or this is an epilepsy region. That data is publicly available, so that can be entered here and might inform when you then go from the top to the bottom of this web page information. And also what we haven't integrated in this version here, but I think it's on the new website, we can also generate reports so that we don't need to go through everything in much detail. You can just type in your area where you have the CNV and the type of CNV, and you get all this information, what I just summarized by looking at the things in a short report. And that can be valuable if you talk to a genetic counselor, for example, or if you just want to, as a biologist, want to have a quick assessment of a region. So, and I think we have implemented this, but I'm not sure if it's in the latest version online already, but we have to do this. So yeah, so again, it takes a village, right? So this is you know, done as a team with the foundations. And just as a sad story is, I wrote like at least 5 grants on this and always completely rejected. So that's completely unfunded work. And that's why our own progress, I would say, is slower than I would like to have it. So we could do more things, but it's challenging. So because the PhD student who was on this project also left, and we have to now build up again. But yeah, other questions?
Thank you, Dennis. That was great. I think we have already a question in the chat. Maybe Nathan, you want to kick us off, and then I would invite anyone else, in particular our AP researchers here, to please unmute and ask a question or make a comment. But Nathan, please kick us off.
Hi, Dennis. Thanks for presenting this. In understanding what you recently showed there at the end of the contour of sort of general population CNV frequency rates across the chromosome, in helping a counselor or anyone else interpret, say, a dip, do you have a measure of just like background sequence repetitiveness to understand how prone to copy number mutations a part of the chromosome might be? And then after that is what happens to those in the population if they harm health or not. But just to kind of know like how much CNV would we expect to see here based on just repetitiveness of sequence at the appropriate scales?
Yeah. To be honest, I don't have a like an expectation distribution, what you would expect, you know, what is default 0. And from the source of data, where we have the data from, which is the UK Biobank, there are also people who have milder forms of developmental disorders. And we don't have the information if, for example, if you see 4 CNVs in a specific region, if that individual has maybe a very mild version of a developmental disorder. But overall, I think the average coverage of a deletion duplication is almost by 1 or 2 individuals maximum, and that's more like error rate across these 500,000 individuals. So it's not a good answer, to be honest. And we don't have the repeat markers why it would break just by chance and why this is a significant number of CNVs in a specific region. But this can be taken from the data, which is some work, but definitely is doable.
So I wonder if even just old-fashioned repeat master tracks from, you know, if that might help a little bit.
Oh yeah, so adding the UCC like repeat track, which can just be downloaded as a bat file. Yeah, makes sense. Yeah.
Awesome. Thanks, Nathan. Anyone else with a question or a comment, please unmute and turn your camera back on as well. Looks like we've got Nicoletta.
Hello, hi, thank you for your talk. I'm Nicoletta Moisoy from UK, and I joined recently the project. So I'd like to ask, you mentioned that you would like to expand the database with data from models, perhaps cellular models, that would correlate genotype and phenotype. And I was wondering whether you have something specific in mind, whether there are any quality control thresholds that would be useful for this type of database, so that type of work that we produce is up to a level that is useful for your site as well as our site. Yeah, that's basically how can we integrate cellular models information into this type of database, and how many patients, for example, how many samples would be useful to have for such database analysis?
Yeah, and to be honest, it really depends on the type of data, and we probably would have to meet and discuss and brainstorm a little bit, and you probably have better ideas than I have. But overall, when you, for example, generate, so the first easiest thing, and I think that's already valuable, is if there is a model available for a specific CNV, if someone would hover over that a region, that it would just be listed or even just linked to a paper. That's the first thing, right? Just saying, hey, we look at this region and actually there's a model available for that particular CNV. Having that information is already a step forward, right? So, because at the moment you wouldn't know, right? So how do you figure that out? Because not every 8P CNV is the same. The second, and that can even be published data, doesn't need to be, unpublished or just contact information. The second thing is, to me, it seems relatively straightforward if one just would generate chromosome 8P level or genome-wide tracks, like transcriptome data or methylation data, and one would just show that how that would look underneath in how this model data looks in that region, that people can just explore that data and are aware about that. It can also be, it could also either one displays this data, or it could also be just a table again, just showing that this data is available and there's opportunity for collaboration or download, depends on the framework.
Thank you.
But I guess to amplify that point, so then being able to, in the portal, be able to visualize, say, images of cells, whether you're looking at, images of mitochondria or other compartments, like being able to just visualize that. And then I guess there's a tabular form or just ways to interact with that data. I mean, is that, I guess, yeah, do you, I know you have to brainstorm more on the specifics, but like just roughly speaking, image-based cell phenotypes and how you might share those most productively.
Yeah, I mean, if the image is available, just pulling them up, making them clickable, that is not so complicated. So just a viewer, we have like 3D protein structure viewers in other portals, right? That's more complicated when you have to integrate these kind of libraries. So I would, so just showing images, that just technically that's very much feasible. And in theory, it would also be feasible to, for clinicians to click on CNV as an identifier and just look at it in EEG or MRI. There are libraries to look at this live, but this comes then to user profiles, data protection, like when it's less, the portal becomes like a research platform with a lot of security around that, when it really goes to this individual level data, and that takes a lot of energy to set that up. So we are moving in that direction now. We have for the single gene disorders, we are moving now to a user account based platform, even with a board where like a Facebook group, but on the portal where your user can log in and communicate with moderators, which are from the foundation, and then on scientists, and then specific more discussions. but we are just moving in that direction. It takes a lot of energy to get this going because it has way more depth in the back end compared to these more short bioinformatic visualizations. But we're moving in that direction.
Thank you.
Great, thanks. Who else has a question or comment? Oh, yes, excellent. Stefan, please.
Hi, Dennis. A very nice presentation. I think you did a great job explaining how genetics can help to make educated guesses. So I had two questions. One was a little bit about the scores that are being used. So a lot of the scores, the haploinsufficiency score, the triple sensitivity score, it comes a lot from comparison against, well, at least the haploinsufficiency score comes mostly from comparison to NOMAD data, right? From a neurotypical population, right? And I'm assuming most of those are single nucleotide variants that affected single genes at a time. So the weight of the score is probably from single genes, right?
The ones from NOMAD, like PLOV, yes, but there's also like a haploinsufficiency score from CNV data.
Got it. Okay, so that was my next point, because I think you're on a preprint with Mike Talkowski and Ryan Collins, and so I've been looking at it because I'm wondering whether in that work you guys found anything that surprised you, because of course with the CNV, I know you know this, just for the audience, so with the CNV, you can get synthetic interaction. Something that wouldn't be, let's say, haploinsufficient on its own might be haploinsufficient if another gene in the same region is also deleted, right? Or duplicated for triple sensitivity. Did you find any surprises like that in your 700,000 CNV cases, where you found something that seemed to be dosage, like an interval that was dosage sensitive, that didn't have any evidence of?
Yeah, because the deletion always hits both, right? And that's why suddenly you never see it individually deleted, but you don't see that in NOMAD with PTVs, right?
Right.
Or you see in NOMAD PTVs. I don't, to be honest, I cannot tell if it's in the supplementaries. I think Ryan, the first author, he looked at this. I think he might have looked at this, but if not, the tables, so the data is available. You know, you could simply join both annotations and see if there are genes, and then ideally you would just make sure that they're next to another. If you then sort by chromosome position, then you would also have directly the answer if there's like, we haven't done this. And at the moment, I don't have someone directly working on this here, so that I cannot just take, because we have also the data, right? But if you extract from the NOMAD, there's a download page, the PLOV scores, and take from Ryan's paper, then also the deletion score, then you have it. You can just do this assessment yourself.
Okay.
But you can compute this, right? And then that would also be an additional, you can flag then these genes and show them also as a separate track, and would make a lot of sense.
Right. And then a subset, so related question is, and I guess one could look for that somehow, is looking for cases where there's a sensitivity to a deletion, but no dosage sensitive genes in that region, but maybe a neighboring region that is a clear candidate for having, being dependent on being dependent on enhancers in the deleted region. I wonder whether you guys have thought about finding ways of tagging those.
Yeah, we haven't. But so let's see. There's a
it would require like a lot of integration, but I looked at your portal, and I've been looking at the High-C portal out of the there's a group that's now at Chicago at University of Chicago that does these 3D viewers. It'd be really cool if you could actually see both, because I felt there were some enhancers or probable enhancers in the distal region of 8p that might actually impact the inverted region. But yeah, so that's kind of where that, where that question came from.
Yeah, I mean, so I assume there will be a lot of iPSC models coming over the next few years with transcriptome data and with different kind of configurations of 8P. And one could then show on the genome-wide scale if there are differences. But that moves and more towards analytics, I guess, than just visualization of regions. But technically, yeah, of course, that would be very interesting. Or you could just show the dysregulated regions and just let them, if you hover over it from which model it comes, you could show all of them. And
right. And then I had a final question. Sorry to be dominating with questions. I had a final question, and that is to go from the, like, I wonder what I tried to, so I talked to Barbara Stranger about this at a meeting once. So I wonder whether you could go the other direction and look at an expression data set, because Nomad, of course, doesn't have it. I don't think has expression data associated with it. But look at an expression data set of, for example, GTEx, and define dosage constraints, gene dosage constraints at that level, because they have 1,000 donors in there. They have like each 20 tissues or so, to find whether, you could come up with an alternative dosage sensitivity score just based on what's observed in a neurotypical or healthy population in terms of gene dosages. Is 1,000 maybe not enough, you think? Is that why GTEx is not being used that way, do you think?
I don't know, to be honest. So the only way we would use GTEx to understand, in alignment, so with the goal to understand brain disorders, is just to see if the lower bar of the expression is still above. I think the individual threshold. I have to look at this so much of this, but that's the only way. So the only thing what I was thinking about, but it's moving towards analytics, and that's even more work, is just taking data from CBio Portal or some other kinds of cancer studies, TCGA or so, and look at all those cancers which have CNVs of chromosome 8 and different kinds of tissues, and see how such if you have, because you have 20,000 or so tumors with transcriptomic data, how these just basically tumor-induced genomic changes influence gene expression and methylation signatures in these tumors. And you could even restrict it to CNS one. You have a lot of noise, but you might see signals that always, if it affects a specific region, then, I don't know, then there are some trans effects or so, who knows. And you might even see that actually for copy number quite a bit, because they might break similar-ish to a 8P CNVs, because at the end the sensitivity for breakage might be still preserved in the tumor, and you see it more frequent. And maybe if you have like a really advanced model, can correct for all the heterogeneity, maybe. But at least you have like a huge data set, and or you could use even data like the DepMap project, where you have all these cell lines which are more homogeneous than, for example, tumors, and where you could also take this data set, use 8P CNVs as anchor and look at their drug susceptibility, if you restrict it to neuronal cell lines, and there are a couple, and because they have tested like 2,000 drugs for each of them, and for each line, and repurpose these kind of data. But it's like these projects, they are highly theoretical, and you need to figure out a way to reduce the noise. So that's why we have never tackled them. But there are massive data sets which could be repurposed and also shown here. So it takes a lot of thinking about how to show them best.
Thank you.
All right, anybody else with a question or comment? We've got about 4 minutes till the bottom of the hour. I want to be respectful of our speakers' time and everyone else who's been watching in rapt attention here. Thanks for those great questions. Anyone from the family side have a question? Feel free also to chime in, and otherwise, I think we can gradually start to wind down here. I guess there's no other questions. Maybe Dennis, any kind of concluding thoughts here? I know we've put out the call now multiple times for data, and that is definitely in the works. I hope hopefully we can get some of this cell data from fibroblasts that's coming down the pike, and then rare base. I think we'll also be supplying some RNA-seq data hopefully soon as well. So I think we've heard the message, data is coming, but any other parting messages or takeaways for us?
Yeah, I mean, let me be bold, and I would say figuring out major, genomic region to clinical representation, correlations, associations. I think that should be feasible as soon as we have like 300 or so individuals where someone on the genomic side spend energy to curate as best as we can the CNV, and if there's like a group of behavioral and epilepsy and developmental scientists who are clinical scientists who then also curate that data, I think that that should be possible, because then we can, I think we should have a fair understanding what are the major groups of disorders, and beyond what Wendy could show in her small paper, which is a good start. But I think we can do just by combining existing data, which is in many health records. We should be more ahead just by what is there, and just I can strongly just advocate for that. That everyone who is here just tries to convince everyone just to share data and health records. I think, yeah, as long as the foundation then takes, and I think you have that in place, that you have like the IRB approval and so on, that you can access that.
Yeah, so it's actually a perfect tie-in. I just wanted to say, the chromosome 8p registry has been revamped, and we're kind of calling it a 2.0 version, but the observations from the clinicians at the hospital in Colorado and the clinic's actually happening this Friday, and I'll personally be there with several other AP families during our retreat this weekend. But nevertheless, that's already kind of cleaned up and organized, and like kind of on a severity scale of observations that the team of behavioral, developmental geneticists, neurologists, epileptologists, rehab, speech, physical therapy. I mean, sometimes those are the most important observations too, neuromuscular. So that is being sorted. Just that your 300 number frightens me, because that is very hard to achieve. So is that arbitrary, Dennis?
It's arbitrary. So if you have perfect data, you might explain it with 10, but probably it's more complicated. But I just say this is arbitrary. That's where we have found for other disorders quite good, associations. But at the end, I feel it's not a large number, and I don't see that. Don't say this to stress you or do maybe in any form disrespectful, right? So it's a matter of our clinical community. If you in the US, if we just would develop an initiative where we transfer health records, and there are ways to do this. Between institutions, and we would have like at 5 big institutions. For example, Cleveland Clinic, we can access 8 million patient health records. And if you would do a search for just 8p, and then the word chromosome and doctor's notes, chances that you have a chromosome 8p is quite high, right? So because who would write 8p and the word chromosome? I'm certain that if you look at 5 large healthcare systems, you find literally 2,000 potential ones, of which then there will be 500 or 400, which are true patients. And then you need to have clinicians who go through the records. So similar what Citizen does, but more in an academic way. And then you have it. But it's just these, if anyone knows a billionaire, I think we could solve so many problems just by brute force in 2 or 3 years. So
I like how you end on that note.
yeah.
Go big or go home.
Yeah.
Yeah. It's good. It's up. I like hearing that, and it's uplifting. So I agree with you. Maybe that's a project that we really put some time and effort behind. And we kind of started that with the Undiagnosed Disease Network in combination with Vanderbilt University too, with a bioinformatician with similar background to you, Dennis. So, we're trying to pilot test this to see what information we can get from an organized health record. And then if there's like kind of value to that, how to scale that a little bit. But I agree with you.
Yeah, so it's a two-tiered approach, right? So on the one hand, you want to go deep. This is what you do from the patient, invitation, rephenotyping at the specific centers with lots of experts. And on the other hand, you want to go shallow but big, right? So, and the combination of both will then probably yield together, will be very synergistic.
Yeah,
Well,
and we have some other investigators, I will say, that actually unfortunately couldn't join, but are also involved in that. We'll have some more data, and we'll probably try to like put our heads together in terms of how to support expanding a tool that you've already put effort and time in behind without grant funding, frankly. So thank you again so much to your team and for this presentation, and for all the families listening in.
Okay.
Thank you, everybody, for joining. This will be available as a recording. We will send that out soon. And thank you again, Professor Lau, for thought-provoking conversation. And thanks, everyone, for the great Q&A. We will follow up with information about the upcoming August roundtable soon enough. But thank you again for joining us here on this rescheduled July meeting, and have a great rest of your day.
Thank you.
Thanks, everyone.